today. We'll be going over the FDA requirements for how to handle OOS and OOT results. There's actually a very helpful and important guidance on the FDA.gov website for handling out of specification and OOT results. And I've taken some of that information and included it in our webinar. We'll be going through phase 1, which is our laboratory phase of the investigation. Here we're going to talk a little bit about when we get that out of specification result, how we handle it, what we should instruct our analysts to do once they get that result, and how to proceed, how to investigate, what to investigate. Then we'll move on to phase 2, which includes a full-scale investigation. That will include not only the laboratory, but also getting our manufacturing colleagues involved, having them start to look at their process, because at this point we are not finding a root cause in the laboratory area. We'll be pulling in the manufacturing group. We'll be talking about what they should be looking at, and a little bit about product disposition at this point. We'll go through a little bit about how to conclude those investigations. We'll also step through a little bit about out-of-trend investigations. Sometimes they are confused with out-of-specification investigations, so we'll talk through those. Some common pitfalls that you may experience during your investigation and how to handle them. And then we'll close out the webinar with some out-of-specification related citations and warning letters. They're very good examples. At this point, they're not extremely recent, but they are definitely relevant to the subjects that we're talking about today. If you get, to a point where, you think, well, I think we've exhausted this topic, we know what we're doing, all you really have to do is look at the FDA.gov website for any of the 483 observations or warning letter citations, even, as early as this year, even last year, every year it seems that there are some observations at least on at least one of the warning letters that you look at related to out of specification investigations and how the company really didn't handle it that well. It goes a little bit, ties in actually with deviation investigations, how to properly investigate, how to make sure that your root cause is robust. So this webinar will kind of tie in all of that information so that you'll not only, report the out of specification results, you will talk about how to investigate it and then how to close out those investigations so that when you do hand it over to a regulator or an internal auditor or a corporate auditor, that they'll understand the logic behind why you did what you did. So what is an out of specification result? We have obviously in our companies testing that goes on in the quality control laboratories. Every one of those tests should have a specification that is assigned to it. The specification is an explicit set of requirements to be satisfied by a material, product, or service. So obviously we have these set up so that our product will actually maintain its safety, efficacy, potency throughout its life cycle. So these specifications are extremely important. They're set up during the early stages of the product development, and they do last with the product through licensure. And then once the product gets licensed, these specifications are included in the license file, and they get included in your actual certificate of analysis when you are about ready to release the batch to the market. In the event that you have one of these specifications fail or not meet the applicable specification, that's when we call it an out of specification or an OOS for short. These are usually seen in the quality control laboratory, although it can be in any really GLP laboratory or GCP laboratory if there are specifications assigned. The FDA does have a little bit on out of specifications in the CFR, section 211.192. They talk about having an unexplained discrepancy or a batch that's failing. So if any of those happen, we need to thoroughly investigate it, whether the batch has already been distributed or not. One thing to keep in mind that I do see a lot of issues with through my consulting career is that a lot of times when companies do come upon an out-of-specification result or a result that has failed, they really just look at that batch that has failed. Now, here in the CFR, they're telling you that your investigation shouldn't stop at just the lot that failed. The investigation is going to extend to other batches of the same drug product or other drug products that may have been associated with this specific failure or discrepancy. So here we're saying that you shouldn't have blinders on. When you have that result, you should make sure that perhaps you look at a few lots that were made prior to the failure and a few lots that were made after the failure and kind of bracket them to see if you have a systemic issue or if it's really just an isolated issue. And as always in our industry, if it's not documented, it's not done. Make sure that you're keeping a written record of what you're investigating, which will include conclusions and follow-up. So here you can find all of that information in the Code of Federal Regulations, Chapter 211.192. When we go through an investigation, regardless of the kind of investigation it is, we're trying to figure out why something happened. When we have an out-of-specification result, you have to have the idea that the source of this result is either an aberration of the measurement process or the manufacturing process. So it's something that should have that can be found in the laboratory or in the manufacturing area. Keep in mind that even if a batch is going to be rejected based on that out-of-specification result, you can't just say, you know what, we're going to reject it. I'm not really going to do a great investigation. I'll say, you know, this happened. This is, you know, an isolated event and just put reject as my product disposition. You really have to still go through that entire investigation. It's very easy to say, well, who really cares? We're rejecting the batch. We're doing the right thing. But we really have to do that investigation because we need to understand if this is just really an isolated incident or if it's going to impact any other batches that may have been made around the same time. We often hear the word robust when we go to these trainings. We need to do robust investigations, robust training. We need to have robust systems. But the only time I really like to hear the word robust is when we are dealing with deviation investigations, especially OOS investigations. When we say something needs to be robust in an OOS investigation, we want that investigation to be very thorough. We want it to be timely. If we get a result that's OOS, we really don't want to sit on that. We don't want to say, well, you know, we got an OOS, let's, you know, we'll let quality know, but, you know, we're really not going to worry about that for a few days. You have to get on that investigation the day that it happens. You need to have an unbiased attitude. I worked in the laboratory for quite some time. I'm a classically trained microbiologist. I've worked on the bench for quite a few years before moving into quality and really utilizing that as the springboard into what I'm doing now. But I always remember being in the laboratory, you know, unfortunately having to report the out-of-specification results and manufacturing and even upper management saying, well, you know, it's the laboratory's fault. You know, it has to be. They probably didn't perform the test properly. Perhaps there was human error. Maybe one of their machines or instruments, you know, wasn't working correctly. Perhaps there was a out of calibration for one of their pieces of equipment. We always were the ones that were the cause initially. So we had a lot of bias around us. Well, it's the lab's fault. Oh, the lab caused an OOS. We really can't be like that. We have to have an unbiased attitude. We have to open our eyes and our minds to see that, you know, it could be the laboratory or it could be something that occurred in manufacturing. Always well document everything that you're investigating, and of course, make sure that you're investigating in a scientifically sound manner. Make sure it makes sense scientifically. Your investigation is going to determine if the result is associated with other batches and if those batches are going to be affected. Again, batch rejections or discarding that batch does not negate the need to actually do the investigation. You still need to do that. As we all know, as I said in the beginning of the webinar, there is the out-of-specification guidance on FDA.gov, but it wasn't always there. Some history behind it, as you probably all know, there was a landmark court case, the USA versus Barr Laboratories, where the inspectors, FDA inspectors, had descended upon Barr Laboratories quite many years ago. They had some issues with the way that they were documenting their out-of-specification results, their additional testing, and they alerted their headquarters. As a result, a lot of things had occurred. They found a lot of egregious issues, a lot of testing and testing and testing with no real endpoint, and it became quite a big deal. They actually went to court, and in court, they actually had over 3,400 pages of testimony and 500 exhibits that showed that this laboratory really had no idea how to handle out-of-specification results. They were averaging their failing results with passing results to meet the specification. They were conducting multiple retests with no defined endpoint. So if something failed, they would just test it again and again and again and again. They were utilizing statistical outlier tests to reject chemistry results. And they had no retest policy and procedure. There was no standard operating procedure that guided any of the analysts or employees on what to do if they came across or an OOS result was generated in the laboratory. As a result of all of this, the FDA decided that they were partially at fault. I would like to kind of believe. And they determined that there wasn't a lot of guidance out there for companies at the time. This was in the late 90s, so they decided to issue a draft guidance for out-of-specification results in September 1998. And this became a very, very important document that we're still using today. Judge Wolin was the judge that had presided over the case, and this quote really struck me. And I thought it was quite important, and it really does send a message, to our lab analysts, when they are testing our very important product. He said that testing lies at the heart of a drug manufacturer's successful operation. Through testing, companies validate their processes and ensure the quality of batches for release. So I don't know about you, but if you have any employees or you are an employee of the laboratory, sometimes you go in, you test your products, you go home. It's really not, it's kind of second nature to us, even if we're managing those laboratory personnel. Not a lot of crazy things go on in the laboratory. We kind of get very, I guess it becomes very mundane and second nature to us when we're working in the laboratory. But this quote kind of really struck me because it's really showing the importance that not only industry, but also, in a legal sense, how important testing is. Judge Wolin was saying it lies at the heart of a drug manufacturer's successful operation. It is ultimately responsible for the release of batches. Without any testing, we wouldn't know if a product was safe, pure, effective, potent, for our customers to take, to ingest, to be injected with. So it's extremely important that we let our analysts know the importance of their testing, that they should really try to maintain accuracy, and that, when they do come across an out-of-specification result, that it's taken very seriously. A little bit about the guidance. It is chemistry-based. It doesn't really talk about microbiology testing, serology testing. It is definitely chemistry-based lab testing. of drugs that are regulated by CDER and CBER. For those of you who aren't familiar, CDER is the Center for Drug Evaluation and Research at FDA, and CBER is the Center for Biologics Evaluation and Research at FDA. It does apply to testing that is performed on active pharmaceutical ingredients, excipients, in-process materials, and of course, our finished drug products. It does apply to products that are purchased outside but tested in-house. Contract firms that may be doing your testing as a contract laboratory should also be following this guidance. The FDA does require that any of your drug products and your APIs are to be manufactured in accordance with the current GMP under Section 501 of the Food and Drug Act. Current GMPs for APIs actually includes performing scientifically sound raw material testing, in-process monitoring, release and stability testing, along with process validation. So if you get any out of specifications in any of those types of tests, FDA is expecting that you're going to do an adequate investigation of those out of specification results. And again, a reminder that the responsibility of a contract testing laboratory in meeting the rules and regulations of the guidance document is really up to the manufacturing firm. So if you as the firm have a laboratory doing the testing for you, you have to police them. You have to make sure that they are following the guidance, because if the FDA goes to that contract laboratory, finds out that they're testing your product and they're not following the guidance, your product is going to be held in question. There are a number of lab results that we can get in the laboratory. First and foremost, as we've been talking about, the out of specification results. Again, something that's not meeting the results in the official compendia or any of the specifications that are developed by company documentation. We also have out of trend results, or OOT results. These are generally found in the stability testing program. Generally, a stability result is not following the expected trend, maybe with other stability batches or maybe with respect to those collected in earlier time points. So, hopefully you all know a stability study is done on products that are released to market. They're tested while they're on the market at certain time points, maybe 1 month, 3 months, 6 months, 12 months, expiry and post-expiry. And if any of those results seem to be a little bit out of trend, that's when you would start an out of trend investigation. Sometimes you may have starting materials or in-process materials that may also yield out of trend data. It may not look like a typical data point, but you still may need to investigate. You may also find out of trend results in your environmental monitoring program for viable and non-viable data. Perhaps you may have an out of trend result or something that may have exceeded the alert level. And there is another type of result. You can call it an atypical, aberrant, anomalous result. But it's a result that's still within specification, but it looks funny. It's unexpected, it's irregular. One example would be a chromatogram that showed an unexpected peak or perhaps an unexpected result for a stability test point. We go now into phase 1 of the laboratory investigation. Before we talk about what to do, let's talk about how you should approach an out of specification result. You have to have a standard operating procedure at your company that dictates how you handle these types of results. Most of industry does it this way. We'll use an example, very easy example. We'll say that Sally is doing a pH test, let's say. Very simple test. We've done them in high school. but there's a specification for this particular product. The specification is that it cannot be, well, let's say it has to equal 7.1. The pH has to be, well, let's make it a range. We'll say it has to be between 7.1 and 7.3 to be passing. So it can be 7.1, 7.2, 7.3. Anything less than that or more than that would be considered out of specification. Sally's been testing pH for quite some time. Her training is up to date. She gets, lot 1, 2, 3. She decides to send it through the pH meter, and she gets a result of 6.9. So it's a little bit acidic, more acidic than we'd like. She immediately consults her standard operating procedure, and this is now called her original result. This is her, original first result. The standard operating procedure says that she can do another test with the same reagents, the same analyst, the same everything, and that would be called her confirmatory result. So she, at this point, before she goes forward with her confirmatory result, she lets her supervisor know that, hey, I got a 6.9. I looked at everything. It looked normal. I'm trained. Don't think it's me, but, we'll do the confirmatory result and see what we get. She goes, another time that she does this batch. She has some extra sample left over. She runs that through the pH meter, and this time she gets another 6.9. That does confirm her original result. At this point, she's done. She cannot test anymore. She cannot, change anything. She has now confirmed that that is an effective, an original, wholehearted, out of specification result. There are some times, though, that you may get, let's say her second test, she got a 7.0, or actually a 7.1, because that we said would be passing. So she has a passing confirmatory result, but a failing original result. In this case, many times I get questions at the end of these webinars and say, well, why don't you just take the passing result as passing? It did not confirm. Why did you go into the, investigation? Well, let me tell you, there's a reason why she got a 6.9 in the first place. We have to figure that out. We have to understand that, even though we'd like to believe that the 7.1 is the correct answer, perhaps the 6.9 is the correct answer. So when in doubt, if you have an original result and a confirmatory result that do not confirm one another, you still need to go through the investigation process. At this point, we're going to start phase 1. We're in the laboratory. We're going to look at all the data. Again, we're making our investigation robust, timely, unbiased. We're going to look at your test preparations, making sure that they are not discarded. We want to make sure that that lab bench somewhat, mimics a, let's say, crime scene. I used to say in the laboratory, make sure that it's kind of cordoned off with that yellow tape when there's an out of specification. You just don't want anything to be discarded or moved or changed in any way, because you want to keep that the integrity of that setup. If you have a contract laboratory, make sure you have a quality technical agreement set up with them where they alert you in the event that this happens in their laboratory. Your own quality control or quality assurance group will work with them on what to do next. They should be starting this phase 1 as well, even though they're a contract laboratory. Your analyst will have a number of responsibilities at this point. He or she should be responsible for making sure that we have accurate laboratory data and results. You want to make sure that they are trained properly on the tests that they're performing. All instruments are meeting proper specifications and are properly calibrated, and the analyst should be aware of any maybe pitfalls that could exist with these tests. What I mean by that is, if they're a little bit sensitive, I had one chemistry test that was extremely sensitive to humidity, so we had to make sure that the room maintained a constant 70% humidity. If it dipped any way, one way or the other, the test would be affected. So make sure that your analysts are aware of those requirements. The supervisor or manager needs to react quite right away and quite quickly. They should be objective and timely. They shouldn't assume that it's not the laboratory's fault or it's automatically the manufacturing area's fault. They should start to look at the data as soon as possible. And they should also start to generate what they think a hypothesis for this occurring could be. So, you know, do I think that it's the laboratory? Do I think that it's perhaps in manufacturing? Do I think maybe one of my lab analysts might have done, you know, had an error? All these things should be going through their minds. They will start to discuss the method with the analyst who performed it, look at the calculations, look at the instruments, the standards, the reagents, and start to prove his or her hypothesis. At this point, after you've looked at your entire process, training, the laboratory equipment, the laboratory reagents, and all of the other things that go along with the test, if you cannot find evidence of laboratory error, you are done in the laboratory. However, when you have clear evidence of a laboratory error, laboratory testing results should be invalidated. So in this case, if, let's say, at the end of our lab investigation, we find that maybe Sally used an expired buffer in the pH test, we can invalidate those results. It was a clear error, and we replace that. We utilize the correct buffer that is not expired, and then we obviously annotate those results. When the evidence of the laboratory error remains unclear, then we move to phase 2, and we get our manufacturing colleagues involved. This part of the investigation may consist of additional laboratory work, additional testing, and of thorough review of the process. The objective of this investigation is going to be, we want to understand why or what caused that out of specification result, because we want to fix it. We don't want this to happen in the future, so we want to do an appropriate corrective and preventative action. We let our QA counterparts know that now it is moving into a phase 2 investigation because we cannot find any clear cause in the laboratory. And QA will oversee the investigation. They don't usually conduct it, but they will oversee it. You can certainly get a group together to help look at the, the actual issue. I always like to get some people who aren't really familiar with the, the test or the process involved. They put a new set of eyes on things. So, maybe have a nice cross-functional group, maybe pull in a maintenance individual, maybe engineering, validation, really up to you, but a nice core group of individuals that can maybe look at things a little bit more objectively. Do a complete review of your manufacturing and process development process development documents with a written record of what you found. When you have, and maybe you have manufacturing that is also done off-site, perhaps with a contract filler or a contract fermenter or formulator, you want to make sure that those sites are involved in the investigation as well. Documents such as cleaning logs, batch records, of course, line clearance logs, training logs, all of those should be reviewed as part of this process. As we sit down, to either handwrite or type up our investigation, we want to start with a clear statement of the reason for why we are doing this investigation. We could say on such and such a date, analyst, and use their initials, let's say SA, performed a pH test, which resulted in a 6.9 and a 7.0, which confirms a true failure for this test. And you can, go on from there. You want to include a summary of the manufacturing process aspects that could potentially cause the problem. I like to do this in the form of a flow chart. It's a nice way to show the process. If it occurs in a formulation area, or let's say in a fermentation area, or in a filling area, it's a nice way to give that, the third-party reader an understanding of your process. Pictures are always worth 1,000 words, so a nice flow chart is a nice graphic representation of the process. Once you have that down, you can kind of highlight where you think something went wrong and then explain it as part of the text. Always review your documentation with probable cause. Be suspect. Always have that, don't really just take it for what it is, kind of question it. Look at previous reviews that may have been done on other batch records for lots that were made prior to the failure. That may help as well. And of course, once you think you found the root cause, you want to make sure that you have a very descriptive corrective action. Much like any type of nonconformance or deviation investigations, out-of-specification investigations need a robust root cause analysis section. So many times I've gotten these across my desk where it says, isolated event, root cause could not be determined. Please don't write that down. I know you know that no one just looks at a deviation and doesn't do a lot of investigation. They look at documents. They talk to people. They look at evidence. They look at swipes from who went in and out. Perhaps they look at video footage of the process. There's a lot that goes into a deviation investigation, so give yourselves credit for that. Don't just write down, I couldn't find the cause, this must be isolated. You may not find the cause, but let the reader know how much you investigated and why you ruled those things you investigated out as a true root cause. Again, create that timeline. Make sure that people know when it happened and how you worked your way to either solving the issue or whatever your conclusion may be. Having that new set of eyes on the issue with your cross-functional team is really important. Making sure that you have a timely investigation, but it's thorough. So that's definitely a tight wire to walk. Most companies have a deviation timeline. Let's say the standard is, let's get all of our deviations closed within 45 days. Perhaps an out-of-specification investigation might take a little bit longer than that. I would rather you exceed your timeline by a few days instead of really writing a poor investigation. And always really look at everything. Leave no stone unturned when you're dealing with this. You may or may not want to do some additional testing in phase 2. You can do retesting or resampling. Retesting will always be testing a portion of the original sample brought into the lab. So much like our example, Sally didn't ask for a new sample for the pH. She had some leftover that she utilized in the second confirmatory test. If you have even more leftover, perhaps you want to use it as part of a retest. Make sure before you do any additional testing other than the original and confirmatory that you get number 1, QA approval. Number 2, you write a protocol that actually outlines what you're doing, how many replicates you're going to test, and what you're going to do with that information. You want to specify the number of retests. So if you're going to test it 3 times or 5 times, just make sure there's an endpoint. Don't test into compliance. This is exactly what got Bar Laboratories into trouble. This protocol of testing will be reviewed by quality and signed off by quality so that they are aware of what you're doing. Retesting is usually performed if you think that you have a testing instrument malfunction, perhaps a sample handling problem, or maybe something happened during a critical task. We like those protocols because it does stop testing into compliance. Testing into compliance is basically the practice of ignoring valid information that should be used to make decisions. It is an unethical practice, it is immoral, it is illegal, it is fraudulent. If you see someone testing into compliance, alert your management. They will need to be stopped. And many times it does result in the employee's termination, unfortunately. Some examples of testing into compliance, as we stated before, averaging out of specification results with passing results to get a passing result. You're physically averaging powers, granulations, and liquids to get in-spec results. Perhaps not recording data until it's known to be in specification, or your samples just basically go missing. I had a situation where I had an analyst who worked weekends. Her one task was to do our water system testing on Saturday and Sunday. She would get quite a number of samples down. We had quite a large campus, so she would get samples from all of the different buildings, and she would have to do, a membrane filtration test on those samples so that we could see what bioburden was existing in that water. She would get probably about 30 to 40 samples, which was quite a lot to do, but she would do them on a Friday or a Saturday. She'd incubate those plates, and then they would be read by the resident microbiologist on either Monday or Tuesday. And every day we came in and, we had passing results. But when I went down to look at the data, they were all zeros. And I said, wow, our potable water is literally zeros. We don't have any bacteria, even in our water that's coming out of our bathroom sinks. That's amazing. What is going on? But when we looked further at it, the analysts that were reading the samples kind of questioned it because they didn't really see the telltale vacuum mark that is usually left on the filter paper when someone is doing a membrane filtration test utilizing a manifold. So for those of you who don't understand or don't have or aren't familiar with this type of microbial test, usually you'll get a 200 milliliter sample. You will take the sample, you will pour it 100 milliliters in a filter cup. Could be a 6-cup manifold. It's just 6 cups in a row. It's hooked up to vacuum. The vacuum will pull the water down over the filter paper. You'll do a few rinses with WFI, and then that filter paper will be removed and placed on an auger plate and incubated for about 3 to 5 days. In this case, we weren't seeing that telltale sign that the vacuum was applied, and then we started to find samples in the trash that were unopened. They still had 200 milliliters of product or water in them, indicating to us that this person was not actually testing anything, because if she did, at least 100 milliliters of the actual sample would be removed. So again, this really wasn't missing samples, but she was testing into compliance, making it look like she was, testing, and she actually was not. Sometimes when we do the protocols, we might think that, something looks a little fishy, and it is testing into compliance. If you decided to do quite a few replicates and all of the data is acceptable, you just report all of that data. It is not, testing in compliance. Neither is failing system suitability. Resampling is a little bit different. This is where you analyze a specimen from any additional units collected. You may actually have to go up and take a new sample from the batch. Make sure, according to 211.165 of the CFR, that there are control mechanisms still in place for examining those additional specimens. So if you're going up into the Class A area to get another sample, let's say in the formulation area, Make sure you're gowning, make sure you're doing gowning plates, make sure you're doing all the EM that you need to, even though you're just up there to get a sample. Resampling really should be a rare event. Your original sample should be enough for retests, and the original sample really must be representative. I don't like going back into tanks or even pulling additional vials. It's just so much better to just retest, even if it's a partial amount that you've already had in the laboratory. I don't like going back into it. It just adds that much more uncertainty that there could be additional contamination perhaps, to the product. Again, with averaging, make sure you are not averaging reportable values. Your quality group must see all of the reportable values, and of course, don't average passing and failing results. With regard to outliers, if you remember, Barr Laboratories actually did use some statistical outlier testing for rejecting some chemistry results. The United States Pharmacopoeia expressly allows you to utilize outlier testing, but only for biological and antibiotic assays. It is silent on its use for chemical tests. But in the court's view, with this, they viewed the silence of the USP with respect to chemical testing and outliers. They assumed that because they were silent on this piece, that they thought it was prohibitory. So in this case, because it's not, mentioned in the USP, the court viewed that it was not allowed to be used on chemical data. If you have biological assays or antibiotic assays where you do want to utilize the statistical outlier testing, again, you'll be writing a protocol determining what test you're going to be doing in advance, the test and the sample size. You will also be able to perhaps reject the biological data. But again, you have to make sure that you're not applying this to the chemistry tests. At the end of your investigation, as you go through all of the different parameters to look into, training, the documents, the equipment, the instruments, all of the different interviews that you're going to be doing, and you're going through that root cause analysis process, and you actually do find a cause, as we said, you can invalidate that initial result and use the retest value in its place, much like I said that, Sally was using the wrong buffer. If that OOS is confirmed, that batch is rejected. You will have times, and more often than not, that the OOS is inconclusive. The retests for your testing protocol may be in spec, so QA has to make that decision as to whether or not they are going to release that batch. So it's really up to the quality unit to determine that. A little on the out-of-trend investigation. We talked about them with regard to stability results. Again, you can have these out-of-trend results in a stability program. You can have them in an in-process or starting material process. It may, again, not look like a typical data point, but again, it is certainly not OOS. And in your EM program, you may have alert levels where you have, a caveat such as this, where if you have 2 or more consecutive samples exceed your alert level, you're going to start an out-of-trend investigation because it's kind of getting close to that action level. Maybe you have an increasing level of bacterial counts. Maybe you have an overwhelming organism that's always identified, such as Staphylococcus. So you have to give consideration to kind of treat those results as, action level excursions, and you need to start to investigate. So out-of-trend investigations are very important to our stability and our EM programs. Laboratory errors should be rare. This is one pitfall that you may find in our specification results. Sometimes you may find that analyst that just, really isn't properly trained. Maybe the equipment is poorly maintained or calibrated. So you may fall into that, that pitfall where you have, additional training to do, or you have to request capital for additional instruments and machinery. But, it should really, be few and far between. Again, keep in mind that do not rush that investigation. If you have a timeline, make sure that you are not rushing just to meet that timeline, that you are still doing a good investigation, and you're not rushing to get it done. Our last part of the webinar, we're going to go through a few citations, as I said before, that kind of bring everything that we talked about full circle. Here we have Dr. Reddy's laboratory. Again, this is part of the warning letter. FDA had said that the out-of-specification investigations did not include analysis of all available data. They gave an example that the inspection revealed that your firm rejected 3 consecutive lots of an intermediate used in the manufacture of an API for the product on February 26th for appearance problems. The deviation investigation into the failure did not include the analysis of all available data, including batches manufactured prior to the failures, which brings us back to the CFR requirement that you must not just investigate the batch that failed, but any of the batches that may have been associated. They also found that they had other deficiencies. They had issues with the timeliness of closing the investigations, the notification of the quality unit of failing stability batches. There were 4 to 8 month delays at this company in reporting failing stability batches. This is very, very serious because those products are on the market when they are being tested for stability, and just basically failing to follow written procedures. The laboratory actually did respond, but the FDA did say that they were still concerned that a thorough evaluation of your investigation approach has not been conducted, so they asked them to do a further, more robust corrective action plan. APP Pharmaceuticals also had some issues. They failed to identify an assignable cause for a failing assay result for their heparin lock flush product. They failed to extend their investigation to other batches, again. In their response, they said that the investigation was completed for the root cause of this out of specification, but apparently they forgot to include it, so the FDA could not evaluate it. They also went on to say that they were concerned about a potentially related trend of out of specification assay results for batches of the API. They asked them for 4 things. They wanted that investigation that they, forgot to include, whether other associated lots may be affected by the issue, whether they had implemented for commercial production the new filter and tubing that was being used when they produced the failing batch. And the biggest one of all is they wanted a 3-year history of this these products that have had out of specification results, all of the testing, the root cause, and the investigation conclusion. Because obviously they were very concerned about the way this company was handling their investigations. So as we come full circle, make sure that if your OOS investigation confirms the OOS result and you are successful in identifying its root cause, you can successfully close out your OOS and reject your product. If your failure investigation extends to other batches that may have been associated with that, make sure that you're including all of that information in your investigation. Out-of-specification results are not fun to deal with, but they are telling us something. They indicate that there may be a flaw in the product or process design. So it's essential that redesign of the product or process be undertaken to ensure reproducible product quality. So that's why it's very important to the company to make sure that these out-of-specification results are taken seriously, the investigations are done, very robustly and thoroughly, and that they are closed out in a timely, compliant manner.